So this is the video for part 2, and we're starting at slide number 10. So if you haven't watched video number 1, you may want to. It's up to you. Starting at slide number 10, module 1 transformation. In the lab, essentially the plasma would have been made for us, and our first step would be to perform chemical transformation. Now if you remember transformation, we learned about it in unit 1 when we learned about antibiotic resistant bacteria. And transformation is where, when we learned about it, one bacteria dies and naked DNA from that bacteria is floating around in the environment. Then living bacteria that are also in the environment and happen to become nearby, they essentially just take in that naked DNA and incorporate that naked DNA from the dead bacteria into its own DNA. So it would then have the traits of the dead bacteria. For our transformation, we don't have to kill off a bacteria. We essentially had that plasmid that we did recombination DNA on. It has the gene of interest on it. And we are trying to force that plasmid into our bacteria. And so it says, how can we get the cells to take in the vector or the plasmid that has the gene of interest? The question is, what charge does DNA have? So hopefully we remember that DNA has a negative charge. We should know that from gel electrophoresis. What charge does a cell membrane have? The cell membrane actually also has a negative charge. And so that's the exterior of the cell. And it says, is a plasmid able to permeate the cell membrane? Well, if the cell membrane has a negative charge, and our plasmid vector DNA also has a negative charge, we should know that the two like charges will repel each other, and so it's going to be hard to get that DNA inside the cell. So we're going to have to do something to help make the transformation happen. So what we would have done is what's called chemical transformation. So you can see here, here's DNA. We have the nitrogen base like adenine, guanine, cytosine, thymine. We have the sugar, and we have the phosphate, which makes up the sugar-phosphate backbone of DNA. And we see here that these oxygens naturally have a negative charge, as we mentioned before. Then DNA is naturally negative charge. So what we would do for step number 1 is we add a transformation solution, calcium chloride, to the DNA. Calcium chloride has positive ions, making it has a positive charge. And so essentially they would find those negatively charged oxygens and they would kind of like bond together. Essentially what they're doing is they're shielding that negative charge. So with the positive and a negative together, it's kind of like this doesn't really have a charge. And they're shielding each other. We would then incubate this fluid with the bacteria on ice, and so it slows down the fluid, it slows down the DNA, everything's essentially going to slow down in its movement because of the cold nature. And then we would do a sudden heat shock. So what that sudden heat shock would do, it says, is it increases the permeability of the cell membrane. So essentially, it's like the cell membrane would suddenly move after being cold. It would allow for some openings or pores in the cell membrane. And it would allow, through those openings, this DNA to enter the cell through the openings of the cell membrane. Once you have the sudden heat shock, or you've done the sudden heat shock, you would then essentially put the bacteria solution in a nutrient broth, aka like bacteria food, and incubate it to allow for the cells to recover and grow on the or in the nutrient broth that is needed. So questions at the bottom says, what happens if you heat shock for too long? What happens whenever you put bacteria in any sort of heat for too long? Hopefully you should know that the bacteria will die. What happens if you don't heat shock for long enough? If you do not go through the heat shock for long enough, then the cell membrane will not have increased movement and will not open, and the DNA will not be able to enter the cell. And so essentially you will not perform chemical transformation because the DNA plasmid is not able to enter the cell. This table is important. I want you to put in your lab journal, even though we're not going to be able to do the experiment itself. This table is really important in understanding how this all works. I'll tell you right now. Okay. So this first column is explaining what you put on a plate, and then you're only going to be filling out this prediction column for now. And so prediction column for growth, you would put a positive if you believe that the bacteria explained in column 1 would grow, or you would put a negative if you do not think it would grow on the plate listed. For glow, same here. Positive if you think the bacteria would glow, meaning it has a GFP protein being expressed and made, and negative if you do not think it would glow. Now, with that said, I want to explain this column first. This first column here says it's an LB plate. So LB plate is just a normal luria broth plate that only has bacteria food on it, and that's it. And you're putting bacteria on this plate that does not have the DNA, it does not have the plasmid or gene of interest. So it's asking, would you expect normal E. coli that does not have this newly inserted plasmid, would you expect for it to grow on a plate that has only bacteria food? You would put a plus sign if you believe it would grow, or a negative sign if you believe it would not grow. Then you'll put like a little slash as seen here. Now we're going to focus on glow. Would you expect E. coli without the plasmid to glow on a plate that only has bacteria food? If so, you would put a positive sign. If not, you put a negative sign. So you kind of can go through all these and do the same thing. And the next plate is saying a bacteria E. coli without the P. fluoroplasmid, but it's on a plate that has luria broth bacteria food. You can kind of assume that. And this plate also has ampicillin. So would you expect an E. coli without the plasmid to grow and or glow on this plate? The next two is referring to bacteria that do have the plasmid inserted. So would you expect E. coli with the plasmid to grow on a plate with bacteria food and ampicillin? And would you expect it to grow and glow there? Would you expect a bacteria E. coli with the plasmid to grow and or glow on a plate that has bacteria food, ampicillin, and a chemical known as IPTG? So you record your results here on the prediction. On the next slide, just to warn you, I'm going to go over the results. So you may want to just do your best to kind of see if you got it right here. It's easy to go straight to the answers, but on your quiz coming up soon, I'm going to give you scenarios. So I want you to understand it more than just know it. So warning, I'm about to go to this next slide that has the answers. If you're not ready, you may want to pause. So here's the results. On a bacteria that does not have the plasmid, would you expect E. coli to grow on a plate that has bacteria food? Yes. Any bacteria, in the presence of bacteria food, should grow. That should not be a problem. Would you expect E. coli without the plasmid to glow? No. The plasmid gives it the properties of glowing because it has the GFP gene, which makes a GFP protein. So no, you would not expect it to glow because it does not have that plasmid. Would you expect E. coli to grow on a plate with LB, bacteria food, and ampicillin? No. Ampicillin is an antibiotic that kills bacteria. if this bacteria does not have the plasmid with the ampicillin resistance gene, which it does not, then you would expect the ampicillin on the plate to kill the bacteria. So there should be no growth. And obviously if the bacteria is not growing, it can also not glow. It does not have the plasmid, so the ampicillin kills it, and it's not able to have the GFP protein to glow. Now in these two, we do have the bacteria that have the plasmid. So would you expect the bacteria that has a plasmid to grow on a plate with bacteria food and ampicillin? Yes. The plasmid allows for ampicillin resistance, therefore it can grow on the plate with ampicillin. Would you expect it to glow? No. The reason is that inducible promoter must have IPTG in order for it to allow for the expression of the green fluorescent protein gene. So since this plate does not have IPTG, essentially the gene is turned off and it cannot be read and it will not glow. So that leads to our last one. Hopefully that last little bit made sense and can help you understand it. On the bacteria that has a plasmid, would it be able to grow in the presence of bacteria food, ampicillin, IPTG? Yes. It has the plasmid, so it can grow in the presence of ampicillin. Would you expect for it to glow? Yes. It has the promoter, and it has a plasmid, and it has IPTG. So it allows for the expression of the green fluorescent gene here, which will allow for it to glow. So hopefully that makes sense. This is a big part of the lab. Let me look real quick. What do we have going on here? Okay. So this is a big part of the lab. If you have questions, I want to put a Google Doc in Google Classroom for you to post questions because I know this assignment's a little bit more difficult. Please construct this journal in your journal, a paragraph with these terms, and hopefully that will help you understand what's going on. Next happening is Module 2 and Module 3. Module 1 is definitely the most difficult, but I would like to go through Module 2, Module 3 with you, but it looks like I'm at 13 minutes on my max 15 minutes. So I'm going to make Module 2 and Module 3 their own video. So our next video, we will start at slide number 15. So if you need help with that, then you can just go to part 3 of the videos.